Hello everyone. In this video we are going to explain the individual drugs. In the past time, we agreed that the previous video would be dedicated to explaining general virology and general terms. We illustrated the viral life cycle, its structure. We pointed out some of the main points in the life cycle that we are going to deal with. Thus, we won't have to explain them again each time we encounter these terms. In this video, we are going to discuss the individual drugs. We will take them family by family. For example, we will discuss the drugs of HIV, then drugs of HCV, HBV, and so forth. And since you understood the past video, then I can promise you that this video and this information will be as simple as could be. The only problem that might bother you is the name of the drugs, because these names can be similar to one another. However, we will make them easier for you somehow. We will help you to remember them, and even show you some alternative ways to get out of the situations when you can't remember them. In the past time, we concluded our video by classifying the drugs in two alternative methods. I told you that pharmacology books are confused between the two methods. The first one is the mechanistic route. We go step by step and explain each drug that can interfere with it. However, I told you this method is of little benefit nowadays and it is no longer used in books. On the other hand, many of the recent books use this method, in which we talk about each family separately, and this is what we are going to do. We will start by the drugs of the HIV, as they are the most important drugs, and they are the ones that demands most of our efforts and our time. Then we will go from the hardest to the easiest, or from the virus that requires a lot of explanation down to the simple viruses that could only require 2 or 3 sentences to understand. So, we will start with the HIV. Then we will discuss the hepatitis family, which are HCV, HBV, and so forth. Let's start with the boss that's called HIV or AIDS virus. Needless to say, it's such an important virus whether you are studying for USMLE, board or whatever. No one would ever pass his exams without being asked in the antiretroviral drugs. Naturally, we explained what does retrovirus mean in the past video. And I told you, it is a recently discovered family. The man who discovered it won Nobel Prize, and naturally, the HIV belongs to the family of the retroviruses or the reverse viruses. This drawing in front of you is the same one that we illustrated and explained in the past video, which is the stages of the life cycle. I will stop at the stages with which I can interfere by drugs. I'll tell you the name of the drug, and only 2 words about each one, which are the only things you need to know. This photo that you see covers all the possible drugs used to treat the HIV. If you want the file, I'll leave a link for it in the description below the video. Print this page and hang it in front of you, and you will never need to study anything more about the HIV. I intended to gather all of the information here on this page, and if we need to discuss a certain detail, we will shift to another slide and then back to this main one. However, consider this page to be all what you need, okay? The antiretroviral drugs. I'll start with the general principles, and then I'll dwell into the specifics or the details. The antiretroviral drugs are classified into 4 classes or 4 families according to the stage at which the drug interferes. For example, we have a family of drugs which inhibit the attachment of the HIV. They prevent the sticking or the binding of the virus to its receptors on the surface of the cells, our body cells, the T lymphocytes. Thus, this family is called fusion inhibitors. You can either call it fusion inhibitors or inhibitors of the viral attachment. What are they? One of them is called enfuvirtide, another one is called maraviroc. But allow me to ask you to delay their explanation a little bit, as they are going to be explained in detail on the following page. But for the start, you should know that we have a family that interferes with the attachment of the HIV, and this family that interferes with the attachment exists only in the HIV. In all of the common viruses, we don't have such a family, okay? This family is specific for the HIV only. Let's move to the next step, the uncoating. In the HIV, we don't have any drugs that interfere with the uncoating. We don't have any drugs for this step, thus we will skip it. However, we still have the large trait, the step of the replication. We agreed that the virus, the HIV, which is a retrovirus, in the past video I told you that the term retrovirus means it uses an enzyme called reverse transcriptase. to make a complementary strand. It is just an RNA, so it wants to form a complementary strand, thus it makes a DNA out of the RNA. It makes itself a complementary copy or a template by the enzyme called reverse transcriptase, which was brought in the toolbox. Then it enters our body cells, the cells that are called T lymphocytes, or the immune cells. It thrusts its DNA in the genome and starts to replicate itself, copying its original genome, then an RNA. Then it sends the RNA outside to be translated into proteins, which are processed by the enzymes that's called protease and so on, the same as I explained in the past video. So we have a family called fusion inhibitors, okay? And we have drugs for these following steps. As we have the second family which interferes with the step of replication. Here we have two types of enzymes. Both are very valuable. They represent a golden chance for us to fight this virus. This enzyme called reverse transcriptase, it doesn't exist except in this family. I can target it. I can target the enzyme called integrase, by which the virus thrusts itself into our genome. I can target this one. However, the third enzyme used by the virus to copy itself, which is called RNA polymerase, unfortunately I cannot target it, nor I can touch it in any way. If you are observant and smart, you will be able to answer me. If you understood the previous video, when I told you some viruses are so malicious that they sometimes use a natural enzyme in our cells, my own enzyme, of our own cells. Thus, this virus, the HIV, after it thrusts itself like this, it uses the RNA polymerase of the natural host cells. It's our own cellular enzyme. Accordingly, we can never touch this enzyme, and that's why I marked it by green color. To remember, we can target this one. However, I can target the reverse transcriptase. I can inhibit it. I can inhibit the integrase. This family is called reverse transcriptase inhibitors. The family that causes inhibition of the integrase is called integrase inhibitors, or the inhibitors of the viral integrase. Then we move to the step of processing. After the virus sends the RNA to be translated, the resultant polyprotein is divided by the protease. I can target this protease and inhibit it by a family called protease inhibitors. Now, if you are watching this video for the first time without watching the previous one, then these concepts would be difficult to grasp, because all of these were explained in detail in the previous video, and we illustrated the role of each enzyme that we are discussing now. Then, now we have 4 classes of the HIV. Let's mention them from the beginning and in order. Drugs inhibiting the attachment, drugs inhibiting the reverse transcriptase, which is this enzyme, the drugs interfering with the integrase enzyme that comes next, and we skip this step, we can't interfere with it. Then we have drugs that are called protease inhibitors. Now I illustrated them in general manner. I just need to tell you about some names of these drugs and teach you some ways to remember these names, if we could, because the difficulty lies here as they are all similar. Most of the antiviral drugs share many letters, thus many of us get confused over them. I told you leave these for now. They are the topic of the next slide. Come here for these. Reverse transcriptase inhibitors. I brought you the most common 5 drugs that you can find in any book. The professor who explains this lesson in your college might add to these 5 1 or 2 drugs, because they are not only 5, they are more, but I'll suffice with these 5, and if you want to add another one, do as you like. But remember, the same principle applies to all of these drugs whatever their number. The first one of the reverse transcriptase inhibitors is called zidovudine, and it is the oldest one of them. The one that followed is called lamivudine. The next one, or the newer in this order, tenofovir. Then we draw a line, and I'll tell you why. The fourth one is called efavirenz, and the fifth is called nevirapine or nevirapine. These 5 are called reverse transcriptase inhibitors, and they prevent this first step, the first step in the replication. Why do we draw a line between the 3 up and the 2 down? Because the 3 up here, which are zidovudine, lamivudine, and tenofovir, their structure is composed of nucleoside analogues. Nucleoside analogues were explained in detail regarding their chemistry, functions, and so on, all in the past video. If you don't remember, go back to the previous video and revise what nucleoside analog means. This is about their composition, and since they are nucleoside analogues, which means they compete with this enzyme, then they are competitive inhibitors. They stop this enzyme through a competition as they enter and compete with the natural nucleoside of your own body. And the viral enzyme gets confused and no longer able to determine which one is the natural and which one is the drug. Thus, it swallows the drug and drinks it, which results in inhibition of the whole step. Efavirenz and nevirapine, which is the newer, and of course there are more, these are called non-nucleoside analogues, which means their structure doesn't look like the amine bases that we have. They are chemically different. And since they are chemically different, then they cause inhibition of the enzyme, a non-competitive inhibition this time. And you can consider these two to be more effective than the previous three. Nucleoside analogues abbreviation is NARTS. N-R-T-S. Or nucleoside reverse transcriptase inhibitors. So these are NARTS, and the others are non-NARTS. Non-nucleoside reverse transcriptase inhibitors. If the names are difficult for you to remember, then I can remind you by a simple trick, if you are a football fan. There was a famous footballer who is called Zainuddin Zidane. So Zidane is the Zidovudine. Zidane was dating 4 beautiful women. These 4 women are Lamia which is Lamivudine, and he was seeing a French woman called Tina, which is tenofovir. The third woman was an Arabic one called Effat. This is efavirenz, and he was also dating Niveen, which is nevirapine. So you can remember them like this. Zidane and his 4 women, who are Lamia, Tina, Effat, and Niveen, okay? And you should know which one of them are nucleoside analogues, which one are non-nucleoside analogues. And what is the difference regarding their mechanism? These induce a competitive inhibition and these are non-competitive. It occurred to me to tell you one last important note. When examiners ask you about any drug whatsoever, either this or the following, they care about the name of the drug, the mechanism, and the third piece of information is if the drug causes specific adverse effect. They may ask about it. You only need to know these 3 pieces of information. Don't distract yourself. The name of the drug, its mechanism, and if there is a specific adverse effect. Okay? Now we know their names. And regarding the mechanism, they induce inhibition of the reverse transcriptase. These are competitive and these are non-competitive. Remember, they all are inhibitors of the reverse transcriptase. At the least, it's not that important which one is competitive and which one is not. What I care most about is if there is a specific adverse effect. They sometimes ask about this zidovudine in medical license exams because it's well known to cause anemia and myopathy. Myopathy means atrophy of the muscles. This player, who is called Zidane, he causes myopathy. This Tina, tenofovir, causes kidney injury, acute tubular necrosis. This one piece of information may be a point in your exam, but only if the examiners hate you very much. They may ask you about this. However, we don't usually ask undergraduate students in it. So, if you are a postgraduate one, be sure it will be in your exams. I'll bring you this tenofovir and ask you which side effect may be caused by this one. Thus, you must remember that this tenofovir causes acute tubular necrosis or at least kidney injury. This nevirapine, the last one, remember that it is the only name among them that has a P. And this P should remind you of hepatitis. Hepatitis means inflammation of the liver, so this P letter should remind you of the word hepatitis. Now it is the time we discuss the next family which causes inhibition of the integrase enzyme. They inhibit this integrase enzyme preventing the virus from thrusting its genome into our own genes of our own cells. What are these drugs? They could be as many as 20 drugs, but I'll tell you about the most well-known two. One of them is called raltegravir. The second one is called ilvitegravir. Tegravir. Their names are already exposed in them. Now take it slowly. If you focus your eyes a little bit now while you are watching me and drinking your cup of tea, you can see that the name of the drug explains the mechanism of its own. So when I tell you tegravir, the word tegra comes from the word integrase, from the word integration. That's why you can remember the mechanism simply by focusing on the name, ilvitegravir, nothing new. Tegra means. From this you should teach yourself the following. Let's assume that I bring you any 4 letters, whatever they are, but they end with the word tegravir. Then they are integrase inhibitors. I'll even tell you something that will help you. This class of drugs which are integrase inhibitors, doesn't exist nor used, except in the HIV. Accordingly, any drug that you may read in any book and it ends with tegravir, then this one is for the HIV. It acts by inhibition of the integrase enzyme. That's it. Nothing more to say. Does any of them have a specific adverse effect? Fortunately no. Only the name and the mechanism. Now let's see the fourth family, which interferes with the protease enzyme that causes processing of the polyprotein and trims it like a tailor. We also have many drugs that could be 10 or 15, however I'll tell you about the most well known two, ritonavir and the other one's called lopinavir, as if one is called Rita and the other is called Lobna. Ritonavir and lopinavir. They all end with the word navir. Again, for the second time, any drug from now till the end of the book, memorize the last syllable, navir. You won't find this word navir except in a drug that acts on the protease of the HIV, not any other virus, because even scientists themselves get confused exactly like me and you, as this field is getting so much bigger. Thus, they decided to standardize the names, so any drug whatsoever that has tegra in the middle, then it means it acts on the integrase enzyme. Any drug that has the word navir, vir means virus. However, this na here, needless to say, is a protease inhibitor for the HIV. If I want to design a protease inhibitor for any virus other than the HIV, then I won't add this word navir. I'll give it another different name. However, if I use the word navir, then it is the one acting on the protease enzyme. Now you know the name and the mechanism. So there is only one item that we miss. Do they have a specific side effect? Yes, and there are questions, difficult questions which are asked in the USMLE very often. The first one, this ritonavir is the most well-known drug all over the ages, the most potent one, to induce an inhibition of the liver enzyme called CYP, which are the family called cytochrome P450. The most powerful drug in all of our curriculum to induce inhibition of the cytochrome and to induce inhibition of the metabolism is this drug that's called ritonavir. And this one piece of information is frequently asked in the exams more than hundreds of times. If ritonavir is ever mentioned, remember that it's the most potent drug to induce an inhibition of the hepatic macrosomal enzyme system. The lopinavir, this last one, it is not very known to cause something specific. However, I saw it very often in the USMLE exams and many other national exams. And the answer is that this lopinavir induces metabolic adverse effects. It causes diabetes, for instance. It might elevate the level of cholesterol or the uric acid. Thus, any mess related to your bodily metabolism is caused by this one, this lopinavir. So this ritonavir causes inhibition of the liver macrosomal enzymes, lopinavir and the rest of them cause metabolic adverse effects. They could cause any metabolic adverse effects. Say if an MCQ stating that one of the following adverse effects is characteristic for this drug called lopinavir or the drug that ends with navir, choose or pick or keep an eye out for anything that's related to the metabolism. Hyperglycemia, hyperlipidemia, lipodystrophy, anything that's related to the metabolism. Now we explained the HIV and HIV drugs. We only have this one family left for the last, which interferes with the attachment, because I want to explain in detail in this following photo. What you see is the virus, the HIV, and this is the T helper cell. This is the HIV, the virus, and this is the T helper cell, which is the most valuable one among all different cells of immunity that we have. This T lymphocyte, the type that's called CD4 or T helper cell. When you study the structure of the HIV virus, you can find that these glycoprotein spikes used for its attachment are of two types. The first one that's colored with red or the orange color is named by the scientists glycoprotein, and they give it the number 120. The number 120 is the result of its molecular weight. However, we don't care much about this. I won't ask you why it's called 120, but at least you should know its name is related to its chemistry. So the first type of spikes is the glycoprotein. That's called glycoprotein number 120. This one in blue color is a glycoprotein, but of another type which scientists call glycoprotein number 41. And also the number corresponds to its molecular weight. Thus the HIV virus attaches to the T helper cell by these two types of spikes. It goes and aims at the T helper cell by its glycoprotein 120 and glycoprotein 41. Where does it attach exactly? When this glycoprotein 41 reaches the T helper cell, it finds a receptor on the surface of the T helper cell that's called CD4. This receptor that's called CD4 is the one that characterizes the T helper cell, so that we sometimes call the T helper cell CD4 cells, which is the most famous receptor on the surface of the T helper cell. So the virus attaches to this receptor by its GP41. Remember them so you don't forget this. It's called GP41. This 4 in 40 should remind you of the 4 in CD4, because there is another receptor on the way. The GP120 also attaches to the T helper cell on another receptor, which is not a very significant one. It's even called co-receptor, which means a helper receptor. It's called CCR5. So remember that the virus attaches by its two glycoproteins to two different types of receptors. One type is major, and the other one is more of a helper nature. It just makes things easier. Okay? Scientists then decided to prevent this attachment using drugs that binds to one of these. At first they thought about the CD4. They said, we can design drugs that block this receptor, thus the virus wouldn't attach. But they failed miserably because the CD4 is the most important receptor, the most valuable engine or epitope on the surface of the cell that's called T helper cell, so we can't block it. When they knew that they cannot target the receptor itself, they decided to target the spikes of the virus instead, this one called GP41, so they invented a drug for the GP41 and called it Antiviral. Where does the name come from? This N in green tells you that the drug doesn't attach to the cell itself, but it attaches to the envelope of the virus. The viral envelope. So they added the EN to remind you, as if they are helping you in exams. Thus the N tells you that this drug called Antiviral, from its name you know it attaches to the virus itself, to the envelope, not the T helper cell. This F letter, this F in the purple color, F reminds you with the number 4. So it attaches to the glycoprotein number 41. This F should remind you of 4, so don't mistake it for another one that attaches to the 120. No, N-Phoviritide. The other drug came to life when they discovered that they are able to block this less important receptor, which is called CCR or co-receptor. We can block it, no problem, my body won't complain. Thus, they invented a drug to block it and called it Mansoura. Where does the name come from? This M should remind you that this drug attaches to the membrane of the T helper cell. So the one that starts with N attaches to the envelope of the virus, and the one that begins with M attaches to the membrane of the T helper cell. And the letter C at the end will remind you of the name CCR5. There are two C here to remind you of the Mansoura. So the Mansoura attaches to the co-receptor that's called CCR5 on the surface of the cell that's called T helper, while the Antiviral attaches to the virus itself, to its glycoprotein number 41. And both of them have the same effect. To use either this one or this one, anyway, you are blocking the attachment. If you want to use them both together, go on, no problem. If you want to use this family in addition to any drug from any one of the previous families, as you like, no problem at all. Whenever you combine more than one drug, each acts by more than one mechanism, you become closer to 100% sure that you are stopping the virus completely. This is the end of the HIV drugs.